Hello everyone, welcome to our channel forensixconceptsforu. In today's video, we are going to discuss about the biochemistry of ABO blood groups and the formation of AB and O antigens as the expression of their subsequent blood groups. So in the previous lectures, we had already discussed about the precursor chain that is present in all the individuals on the surface of the RBC. This is what a type 2 precursor chain looks like, as I have discussed in the previous videos. These are attached, the biomolecules are attached with each other by beta 1,4 and beta 1,3 linkages. So when the H gene is present in the pure dominant form or the hybrid form on the 19th chromosome, it codes for the enzyme L-fucosyl transferase, which causes the addition of L-fucose to this precursor chain and ultimately gives rise to the H antigen. Now this H antigen is the base acceptor molecule which subsequently accepts the further sugar molecules, giving rise to the AB and O antigens of the subsequent blood groups. Without the addition of L-fucose to the precursor molecule, it is not possible to get any of these four blood groups, and this condition, as we had already discussed in the previous video, is known as the Bombay blood group, which is seen in case of pure recessive H gene, which is very rare that it is seen in one individual per 4 million in a population. So what is happening or what is causing the addition of these genes to the precursor molecule or the H antigen base? Before that, we need to understand the gene which is controlling the expression of ABO blood group. The A, B and O variants are of the I gene, which we had already discussed is present on the 9th chromosome somewhere around this area, and the A and the B genes are codominant over each other. Codominance means when A and B genes are present together in the genotype of an individual, they give rise to a third type of phenotype. So the phenotype of the blood group which is produced when both the A and the B genes are present, that genotype and phenotype would be an expression of combination of these two and would be completely different from the expressions of these two genes when they are individually present, and in this case we get the blood group AB. So the formation of this antigen we are going to discuss later. Right now we need to understand that these A, B and O variants of the I gene, they are polymorphic alleles which are coding for enzymes that create blood group specific antigens. That means they are creating antigens which are specific to the blood group of which they are expressions. So these genes are autosomal in nature, that means they are present on the autosomal chromosomes, and the B variant was formed as a result of 7 nucleotide substitutions in the A variant, and 4 of these substitutions, which are the ones expressed here, are 176G, G2355, F266M and G26A. These 4 substitutions caused the coding of an altogether different polypeptide, and as a result of which a completely different enzyme was produced and a third type of genotype and phenotype was seen, which was different from the H antigen and A antigen, right? And the O variant, which is the fourth type of variant, was caused as a result of deletion at the 261, which caused a frame shift mutation, and this frame shift mutation subsequently caused a termination of the translated polypeptide. Now, this termination resulted in the formation of such an enzyme which was inactive. That means it did not catalyze any reaction or any addition of substance to the H antigen base. The A blood group variant is the variant of I gene. So the form of gene which expresses the A blood group is either IAIA or IOIO. So these are the genotypes for the A blood grouping. These genes, they code for an enzyme N-acetyl D-galactoamynyl transferase. This enzyme catalyzes a reaction which causes the addition of a substance called N-acetyl galactosamine to the H antigen, resulting in the formation of A antigen. As you can see in the image, this is the precursor molecule that we had studied about. This is the L-fucose, which is being added due to the presence of H gene, and it ultimately forms the H antigen, and to this N-acetyl galactosamine has been added to give rise to this A antigen, and when this structure gets added, we have the A blood group of the individual. So next we are going to discuss about the formation of B blood group. So in case of B blood group, the variant of the I that is present is IB, or IBIO. This I have mentioned here, and these are the genotypes for the blood group of B, and they code for an enzyme which is called D-galactosyl transferase. So the B enzyme or the B variant is catalyzing a reaction which adds D-galactose to the H antigen base. So this B antigen is nothing but this precursor molecule to which the L-fucose has been added to give rise to H antigen in the presence of H gene, and because of the enzyme D-galactosyl transferase, because of the presence of B variant of the I gene, this D-galactose is being added to the H antigen base, giving rise to B antigen, which ultimately is being expressed as B blood group of the individual. Similarly, in case of AB blood group, the variant which is present is the IAIB form. So this is the genotype for the AB blood group, and it codes for both the enzyme N-acetyl D-galactoamynyl transferase as well as D-galactosyl transferase. So these two enzymes are both catalyzing the addition of two molecules to the H antigen base. This is N-acetyl galactosamine and D-galactose, and ultimately what is happening is, to this precursor chain where L-fucose has been added to form the H antigen in the presence of H gene, both D-galactose and N-acetyl galactosamine is added, giving rise to the AB antigen, which subsequently expresses as AB blood group of the individual. Now, an interesting thing is seen in case of the O blood group. In case of O blood group, the variant that is present is the IOIO, and it codes for a protein substance which lacks any enzymatic activity. So it is not catalyzing anything. The product of the coding of the IOIO gene is not catalyzing any reaction, and as a result of which no addition to the H antigen takes place. So we can say, or we can see in the diagram on the right, that the precursor molecule is having L-fucose added to it, which is because of the presence of the H gene, and because of any non-functional protein molecule produced as a result of frameshift mutation in the O gene is resulting in non-addition of any substance to this precursor and H antigen, and ultimately it is indicating to the fact that O blood group is nothing but H antigen without any modification. O antigen is equivalent to the H antigen because of the production of non-functional protein substance, indicating that O group or the O blood group is nothing but the modified H antigen. So if you really liked our video today, please like, share and comment, and do not forget to subscribe to our channel. Thank you for watching.